Hey guys, welcome back. Today's topic is gene therapy. This is from class 12 biology. Here, gene therapy is a collection of methods that allows correction of a gene defect that has been diagnosed in a child or embryo. So, this gene therapy is allowing the correction of a gene defect that has been diagnosed in a child or embryo. Okay, so here genes are inserted into person's cells and tissues to treat a disease. I repeat this once again. Genes are inserted into a person's cells and tissues to treat a disease. Okay, so this is how gene therapy is performed. Okay, now if we go into the details, the first clinical gene therapy was given in 1990 to a 4-year-old girl with adenosine deaminase deficiency. Okay, so the enzyme adenosine deaminase deficiency leads to a disease called SCID. Okay, this is severe combined immunodeficiency. I repeat this once again, severe combined immunodeficiency. Okay, so this is SCID. Okay, due to the deficiency of adenosine deaminase enzyme, okay, this disease is caused SCID. Okay, severe combined immunodeficiency. Okay, so here the first clinical gene therapy was given in 1990 to a 4-year-old girl with adenosine deaminase deficiency, and here the disorder is caused due to the deletion of the gene for adenosine deaminase. So this enzyme is a protein, so obviously there will be a gene which codes for that protein, and here deletion of that gene which codes for adenosine deaminase will cause its deficiency, and it is resulted in a disease SCID. Okay, so this is one of the very important aspect for exams. So when was the first clinical gene therapy was given? It was in 1990 to a 4-year-old girl with adenosine deaminase deficiency. Okay, now moving to the next. Here ADA deficiency can be cured by bone marrow transplantation in some children. Okay, so this is one of the strategy followed, and enzyme replacement therapy in others. Okay, so these two strategies are followed, but here both of these approaches are not completely curative. Okay, so what are the two methods followed? Bone marrow transplantation in some children and enzyme replacement therapy in others, but both these methods they are not completely curative. Okay, so the other methods to treat this ADA deficiency, let's discuss about them. Okay, here in the treatment of ADA deficiency, lymphocytes from the blood of the patient are grown in culture outside the body. This is in vitro. Okay, so here lymphocytes are grown in a culture medium outside the body, in vitro conditions in laboratory. Okay, so here these lymphocytes, okay, they are produced in large numbers. Okay, a functional ADA cDNA is then introduced into these lymphocytes. Here cDNA for complementary DNA. Okay, so this is complementary DNA. And here complementary DNA is the DNA which is synthesized from single stranded RNA. Okay, this is synthesized from single stranded RNA, and this is often used to clone eukaryotic genes. Okay, used to clone eukaryotic genes, okay, in prokaryotic cells. So this is the importance of cDNA. cDNA stands for complementary DNA. It is synthesized from single stranded RNA, and cDNA is often used to clone eukaryotic genes in prokaryotic cells. So this is how a functional ADA, that is nothing but adenosine deaminase enzyme, okay, a cDNA, okay, using a retroviral vector. Okay, retrovirus is used as a vector, and this functional ADA cDNA is introduced into these lymphocytes which are cultured in vitro conditions. Okay, now such lymphocytes are injected back into the patient's body. So these are the steps followed in gene therapy. Okay, so first of all, lymphocytes from the blood of the patient are grown in culture outside the body in vitro conditions. After that, the functional ADA cDNA is then introduced into these lymphocytes by using retrovirus vector. Okay, after that, such lymphocytes are injected back into the patient's body. So this is how the gene therapy is performed. Okay, now after this, the patient requires periodic infusion of such genetically engineered lymphocytes. This is because, okay, these lymphocytes are not immortal. Okay, these lymphocytes which are introduced into the patient's body, they are not immortal. Okay, they are not immortal. So that is the reason they have to be again introduced. They are not immortal. The lymphocytes which are introduced into the patient, they are not immortal. So periodic infusion of such genetically engineered lymphocytes is very much essential. Okay, however, if the gene isolate from marrow cells producing ADA is introduced into the cells at early embryonic stages, this is before childbirth. Okay, before childbirth, during the early embryonic stages, if they are introduced, then it will be very much beneficial for the child. It could be a permanent cure. Okay, so if it is introduced before childbirth, okay, there is a possibility that this can give a permanent cure for this disease. Okay, I repeat this once again. The patient requires periodic infusion of such genetically engineered lymphocytes because the lymphocytes which are introduced are not immortal. Okay, and if the gene isolate from marrow cells producing ADA is introduced into the cells at early embryonic stages, that is before childbirth. Okay, this is what we call parental gene therapy. Before childbirth, if this gene therapy is performed, okay, then it could be a permanent cure. Okay, after this, now based on the topic that we have discussed, let's discuss some previously asked questions in exams. Okay, so dear students, check your performance in previous year questions. Okay, question 1. Which of the following is commonly used as a vector for introducing a DNA fragment in human lymphocytes. I repeat the question once again. Which of the following is commonly used as a vector for introducing a DNA fragment in human lymphocytes? Option 1, X phage. Option 2, TI plasmid. Option 3, retrovirus. Option 4, PBR322. Here the most appropriate option is option 3. Okay, so retrovirus is used as a vector for introducing a DNA fragment in human lymphocytes. Okay, after this, moving to the next question. Here, which kind of therapy was given in 1990 to a 4-year-old girl with adenosine deaminase deficiency? Okay, here option 1, gene therapy. Option 2, chemotherapy. Option 3, immunotherapy. Option 4, radiation therapy. So here, for the treatment of a 4-year girl with adenosine deaminase deficiency, gene therapy was given in 1990. So here the first option, gene therapy, is the correct option. Okay, now moving to the next question. The first clinical gene therapy was given for treating option 1, diabetes mellitus. Option 2, chickenpox. Option 3, rheumatoid arthritis. And option 4, adenosine deaminase deficiency. So here the fourth option is the most appropriate option. The first clinical gene therapy was given for treating adenosine deaminase deficiency. Okay, now moving to the next question, question number 4. What might be an advantage of beginning gene therapy prior to birth? So prior to birth, before childbirth, or the parental gene therapy. So what is the advantage of this? Okay, option 1, this would give the body plenty of time to utilize the new genes. This is wrong. Okay, next, the body would not reject it as it has not yet recognized self. Okay, so this can be the possible answer, but let's check the other options. Okay, the cells being extremely young are more receptive to gene therapy. No, this is wrong. Next, probably there is no advantage. This is wrong. Second option is the most appropriate option. Okay, the immune system destroys cells expressing the newly inserted genes, but the parental gene therapy that we discussed here, okay, the parental gene therapy, okay, or gene therapy which is prior to birth, okay, it overcomes this problem as the body does not reject them, okay, because recognition of self and foreign is not developed at this stage. Okay, so recognition of self and foreign is not developed at this stage. Okay, so because of this it is advantage. Okay, so parental gene therapy is providing the advantage, or the gene therapy prior to the birth of the child is providing the advantage. Okay, now moving to the next question. Match the following columns and select the correct option. Here, A, Bt cotton, B, adenosine deaminase deficiency, C, RNA interference, and D, polymerase chain reaction, that is PCR. Okay, in column 2, gene therapy, cellular defense, detection of HIV infection, and Bacillus thuringiensis. These are given. Now let's match these options. Bt cotton is associated with this, okay, Bacillus thuringiensis. So A should be 4. So A is 4 here, and in the other options it is not given. So directly we can get the option like this. But anyhow, let's check the other options. Okay, adenosine deaminase deficiency. So this is related to gene therapy. So B should be 1. This is correct. And RNA interference is related to cellular defense, and PCR is for detection of HIV infection. So the first option is the most appropriate option for this question. Okay, so this is all about today's session. If you are new to my channel, subscribe for more sessions. Okay, if you like this, share it with your friends. I'll be back with more sessions. Have a